Okay.
So, my name is Jeff Otto. I work in the area of personalized medicine. I've worked for a number of biotech startup companies. That's basically my background. And about 4.5 years ago, I was recruited into the 5th largest health system in the United States to start up a Center for Translational Research. I'm going to be giving just a bit of an overview on the field of personalized medicine and kind of where we are and some of the challenges that we've seen, and hopefully it'll resonate with folks here. Okay, so I want to just introduce some terms because I understand that probably you guys aren't of the same background as me, so some of these things might be a little bit different. Take a look at some of the early days, take us through the current state, and look at the gap that exists from the early promise of personalized medicine, where we are today, and try to identify some of the things that have impeded the progress. And then I'll finish with a short description of sort of how I'm approaching this now as trying to address these issues. So I'm not going to read through all of this. I actually looked at this and I'm like, okay, let's come up with shorter stuff. So personalized medicine, a very common way to think about this is the right drug to the right patient at the right dose at the right time. Biomarkers, basically these are surrogates for biological processes, whether or not they be natural processes, healthy states, or disease states. And translational research can be really viewed as bench to bedside research. I've also heard it used as bench to bedside to bench. The idea that it's a circular process. So this comes, these items come from the Personalized Medicine Coalition website, and just kind of identifying some of the aspects of the promise and the challenges. And actually, this is pretty timely because I don't know if you guys have been following the news right now in the Supreme Court. There's a case in front of them between Myriad Genetics and ACLU, and the big question there is whether or not genes can be patented. I don't know which way it's going to go, but obviously this is something that would have a huge impact on our industry, actually regardless of which way it goes.
Well, is it a challenge that you cannot patent protect, or is it a challenge that you
Either way, it's going to be it's a challenge because it's unclear, because you have companies like Myriad that say, look, I spent tens of millions or hundreds of millions of dollars developing a particular product. It's very important that I be able to protect this. On the other hand, there's a lot of diagnostic laboratories that exist throughout the markets that say that our inability to offer a Me Too test or to do something like this actually hurts patients. And so there's a lot of people on either side of the issue. Personally, I think that if you're going to be spending tens of millions or hundreds of millions of dollars developing something, you should be able to protect it. But, that's my opinion, and clearly not everyone shares that opinion. So in this cartoon here, one of the key aspects of things is that typically in medicine, people are not distinguished as individuals. I mean, you go into the doctor, you have something that's wrong, you got a flu or something, and they're going to give you a drug that's hopefully going to make you better. And that's going to be largely the case regardless of who you are or what your background is. And this is even though as populations we're all actually very, very different. And so the idea of personalized medicine is that you'd be able to use biomarkers or other tools to help stratify patients into separate groups. So in this case, what you really want to do is that you really want to try to identify this particular quadrant. You want a therapy that's going to be effective and non-toxic, and you also want to exclude this particular population here as well, the idea of ineffective drugs that are toxic, things that will do harm. And so that's really, this is the objective of personalized medicine. I like analogies, and I'm glad we had a nice shoe analogy in the last piece. And so I kind of look at personalized medicine, and the way that we're approaching this is very similar to shoe manufacturing. And this is that a drug company is not going to create a specific tailored drug for you. Instead, what they do is like the shoe manufacturers, they create a product, a drug, that will fit within populations. They understand the size, the shoe manufacturer understands the size of the feet and the distribution within a population. And that's really how we're looking at drugs being developed for populations. You then apply a diagnostic, in this case, a shoe sizer, that's going to tell you what size foot you have, and then you find the shoe that fits you. It's going to feel like it's tailored for you, but in fact, all it is something that was built for a population. You happen to be in that population that shoe fits. And so the idea with personalized medicine is that you end up with a drug that is more likely to work and less likely to be toxic in the population that you happen to exist, and this results in a more effective therapy. Okay, so I've already talked about biomarkers and diagnostics, and one of the key things here is to recognize that biomarkers aren't necessarily these fancy molecular tools. These can be things that have been around for a long time that we're all fairly familiar with. The important thing is that it's just something that serves as a surrogate to help identify some other process. Now, as it turns out, these are from an individualized perspective. These are fairly crude tools. You don't get a lot of insight into what's going on biochemically in an individual from these sorts of tools. So this takes us to my field, which is in the area of, I'm actually a genomic scientist, what we call omic biomarkers. And so omics is something that we've just started coming up with the last few years. That's because people got tired of saying genomic, transcriptomic, proteomic for DNA, RNA, protein, and now we just call it omic. Okay, well, I was going to have a nice video here because I really liked this. So let me give you the story of what I was going to show. This is a press conference that happened in the spring of 2000 with President Bill Clinton announcing that.
I'm going to go over to see if I can find it on each one.
Okay, well, it's a 40-minute press conference, and I only wanted to put it in there.
Oh, so you clipped it out?
I didn't clip it out, but it was at 4 minutes and 45 seconds.
Okay.
And what they're doing is they're announcing, actually prematurely, that they have successfully mapped and completed the human genome. And this really launched a period of, to use Alan Greenspan's term, irrational exuberance. And all kinds of money started pouring into biotechnology companies. If you were doing anything that was related to personalized medicine and genomics, money was just being thrown in. Stock prices were just going absolutely through the roof. And everyone had this view that genomics was going to cure medicine, it was going to cure disease, it was going to do all this stuff, and it was going to happen really.
Who speak about it? This is,
Well, that's Francis Collins. Yeah, but
you said
I'm more. Yeah, he's the head of NIH.
Human genome announcement at the White House. Look at that.
4 minutes, 45 seconds in.
Offer the complexity, the beauty, the wonder of God's most divine and sacred gift. With this profound new knowledge, humankind is on the verge of gaining immense new power to heal. Genome science will have a real impact on all our lives, and even more on the lives of our children. It will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases. In coming years, doctors increasingly will be able to cure diseases like Alzheimer's, Parkinson's, diabetes, and cancer by attacking their genetic roots. Just to offer one example, patients with some forms of leukemia and breast cancer already are being treated in clinical trials with sophisticated new drugs that precisely target the faulty genes in cancer cells, with little or no risk to healthy cells. In fact, it is now conceivable that our children's children will know the term cancer only as a constellation of stars. But today's
Yeah, that's it. I like that.
Yeah.
Constellation of stars.
Yeah.
And the world went nuts. And so, so shortly after these kinds of studies, this was in all the popular science press that even made it to the cover of Newsweek. At the time, I was working at a company called Renaissance Pharmaceuticals, and this gentleman here, Jerry Bovis, was my boss. And I'm not calling him out because he was wrong, but I think that this is, he says, "We strongly believe that pharmacogenomics will shortly transform the way drugs are developed, marketed, and prescribed. I think you're going to see the benefits of this occurring within a 5-year time frame." This is in 2001. And Jerry wasn't unique in that view. I think we all thought that. I thought that. And that really hasn't been the case. And there's a number of reasons for that. At Renaissance, what we found was there was a problem which I don't know if this is a common term, but it's one that we use. We called it chasing the tail. This a term anyone else knows? And so chasing the tail is that if you take a look at a population, a frequency in a population, and most of the population is at this end. And these are people that either, in the case of this drug, either don't respond or are moderate responders. And then there's a small number of people that are that respond very well. So the idea at the time was, well, you can identify biomarkers, you can identify things that are going to be able to help distinguish the people that really respond well from the rest of the population. The problem is that unless the health economics work, it doesn't matter. So yes, you can do this, you can find things that are statistically significant and scientifically valid, but from an economics perspective, don't matter. And so you can do this, but you end up with a product that no one wants. So now just to change a little bit here before I go on to some of the other things, let's take a look at some of the milestones in personalized medicine. So everyone knows about Watson and Crick, 1953. That's kind of when this became possible.
Who knew that already?
In 1977, Fred Sanger developed Sanger sequencing. This later became known as capillary gel electrophoresis sequencing. The cytochrome P450 genes became known to be involved in drug metabolism. The Human Genome Project was launched. In 98, we had our first personalized medicine drug, Herceptin, for breast cancer, along with the diagnostic to go with that. We noticed that the video I showed was in 2000 with Francis and President Clinton talking about how it was complete, but it actually didn't get completed 3 years later. That's because they wanted to get ahead of Craig Venter and Solera and announce it first. Then we had a number of, more recently, a number of new targeted therapies that have kind of come out in the last few years. This slide here, and there is a test at the end, so I expect you guys to pay a lot of attention here, is looking at a cell and the variety of different pathways within a cell that are prone to oncogenesis. And what we see is that there's basically every pathway that's involved, there's at least one and sometimes multiple drugs that have been developed or in the process of being developed that target these pathways. And one of the things that's, I think, really important to keep in mind as we're thinking about personalized medicine is one is these being targeted, that this makes it personal for that particular tumor cell. In addition, there is natural variation that exists within us that also will impact, even if you have a particular pathway as to whether or not that drug is going to be effective in you. So this takes us to here in the question of why so few success stories. You know, one is that the field isn't that old. You know, it's really, make an argument 15 years, 20 years, but certainly not much older than that. Secondly, as we showed at Genescience, technology on its own isn't sufficient. You can develop things, you can do the science, you can do all the work, and that by itself isn't necessarily going to get you to a product that works. I love this cartoon. When I came across this, I laughed and I laughed and I laughed because this is absolutely true. And what you see here is a big haystack of genetic markers. When I came on to CHI and I started doing what I'm doing, a number of people said to me, why are you guys going to go do anything with trying to develop and find new biomarkers? Don't you know that there's already a mountain of them? You know, you can, and the problem is, and that's what's really shown here, is that the biomarkers aren't necessarily fit for purpose. The work that was done that resulted in the identification of the biomarker wasn't necessarily designed for the therapy that it's trying to be used for. And so the challenge really is for the, I don't know if this guy's a scientist or a doctor, but he's trying to figure out which of these biomarkers are going to be appropriate for what he's trying to do. And in all likelihood, none of them will be. Because it wasn't developed with that in mind. It wasn't developed with the end in mind. Another area where people, and I've worked at 3 personalized medicine companies before my current role, and none of them actually knew this, is that the test needs to work within the existing healthcare workflow. And actually one of the best examples I can provide of this is that there's 2 companies out right now with a prognostic for breast cancer reoccurrence. There's a company called Genomic Health. They have a product called Oncotype DX. There's another company called Agendia with a product called Nanoprint. The Oncotype DX, or I should say the Nanoprint test, I think from a scientific perspective is probably a superior test. Science, I think, is better. It's a more robust test, and it's pretty much a failure. And the reason is that the Oncotype DX test works within the existing hospital workflow. It doesn't require anything extra and special of the people within the hospital for that test to work. The Nanoprint test does. It's outside the workflow, people don't want to do it, the test struggles. This actually takes us to the next point here, stakeholder alignment. You know, think about within healthcare all the different parties that have a piece of this. You know, you have the drug developers, you have the diagnostic companies, you have the device companies, you have the hospital administrators, the labs, the pathologists, the physicians, you have the nurses, you have the patients, you have the insurance companies. In order for something new to come in, you need to be considering how does this impact all of these different levels, all these different players, and are they aligned in this? Which leads us to the last point. You know, one of the questions, you know, in running businesses, you know, we're often, you know, who's the customer in this? Who's the customer? Who's the customer in healthcare? I don't think it's such a simple question. But one of the things that's coming out that I think is really kind of interesting is the area of consumer genomics. So what you can do today is you can take a mouth swab and go out and get a profile of a million different traits, or even get your genome sequenced or what have you. I actually, I'm personally more skeptical of the value of these things. So please don't run off and do this and say Jeff told me to do it. But what I think is valuable is that this is starting to put power back in the hands of the patients when they're talking with their physicians. They can say, what about this and what about that? And so we can start to drive the decision making in a way that's more favorable for the patient. And so the more education that we get, the more aware that we are of what's going on and what's available, I think is going to be very helpful to the field. I mean, watching the evening news, you don't see lots of things about diagnostics, but for everything from low T to who knows what else, there are a million commercials on. That the purpose there is to educate the patients so that they're having discussions with their physicians. All right, so now I'm going to switch and just take the last few minutes and talk about my work at the Center for Translational Research and how we're trying to address some of these gaps.
Jeff, what is translational research? What does that mean?
So for us, it is looking at ways to take things directly from the bench and getting this into the patients, getting this to the bedside. So it's the bench to bedside, and then for us also back to the bench from there. All right, so this is a slide that just kind of shows who we are. We're the 5th largest health system in the United States, 81 hospitals across 17 states, or 85,000 employees. We do about 11 billion in revenue. Within CHI, there's an Institute for Research and Innovation, and within that institute, there are 3 centers: the Center for Translational Research, which I run, Center for Clinical Research, which is essentially the group that runs clinical trials, and the Center for Healthcare Innovation, which focuses on population health management. This is we call this the research onion, and this is how we view research within the health system. And you notice that we've identified all of the stakeholders that we think are involved in this thing over here that's called OneCare. This is our comprehensive $2 billion initiative to link the electronic health records across all of these hospitals and basically create a mineable resource for big data. We then use these resources, work with other partners in government, academia, and industry to help define and refine research projects, which we then run. So this here is just another slide kind of focusing on the OneCare piece. So we have our hospitals up here. We have the combined EHR resource. And then right now with my sister organization, the CCR, we're working on the development of a research data mart, which consists of extracted data from OneCare and then with additional annotated data from various research sources. This slide here is just to provide a quick snapshot of some of the different things that we think are important in running these sorts of research questions. This slide here is just to show the overall workflow that we view. And the important thing is that the research questions can come from a number of any different sources. We then take those questions, look at them in terms of the research data that we have. I'm a big believer in doing your math first, and then you go do your wet work after that. So this is part of the doing your math, generating your hypothesis, and then at that point we look to test these within the research laboratories, ideally with the idea that at the back end you end up with results that will have benefit to the patients and benefits to the hospital system. And so one of the questions that I often get is who pays for a lot of this stuff? Yes.
What do you mean by do your math up front? What does that actually mean?
So for us, that means we're looking at the health economics. Okay, so if I'm going to do this up front, I'm going to start on a study, and I know what I hope to achieve at the back end. Is it going to be meaningful?
So we might call it a business case.
Yeah.
Is that roughly
Yeah,
what
it's,
you're making?
yes.
Okay, thanks.
Yeah, so at the back end, everything here, there needs to be an ROI. and the ROI needs to have benefits to the patients. Hopefully that they're getting better treatment, they're well longer, they're well sooner, they have less adverse events, and benefits to the system in that they're not incurring costs that they wouldn't otherwise incur. So if you have someone that's coming in for repeated hospital stays and these things tend not to get reimbursed, that's a problem for the system.
So you would only generate a hypothesis and even pursue it after that generated hypothesis if the research suggests a statistically good probability that there could be a hypothesis that could be generated for a certain mass market.
Well, yes.
Otherwise, if the mass market's too low, then the cost
So what we'll take a look, and a way to think about this is that you identify an area where you think that there's a great area for improvement in the standard of care, and that then I apply the filter and ask, okay, can the tools that I have available to me be used for something like this? Okay, so that's kind of where we get started. Then the next step is, okay, you start looking at your data and asking whether or not you can identify all the variables that are important that impact that, and that if you're successful, can you create a model that would show that if we do treatment course A versus treatment course B with patients, that there's going to be a benefit there to the system or benefit to the patient that will be in excess of the costs incurred to go through the project.
So this is so you don't have chasing the tail.
This is so you don't chase the tail. And so this is kind of an outline on how we try to approach this. So we have our sample, we have our EHR data. So this is basically the complete, medical records of the population that you're looking at, all of the physician notes, the pathology reports, the pharmacy data, what have you. We try to look at things across a number of different technologies. I call this a hypothesis-free approach. So we're not making assumptions about what is going to be the best technology to be applied for the study. Looking at genetics are very stable. Epigenetics are moderately stable. And then environmental is things like whether or not you had broccoli with lunch today or had too much to drink last night. These things all impact how you're going to respond to different medicines. And these are things that can be analyzed. We apply sophisticated biostatistical tools at the back end, hopefully resulting in a predictive signature that allows to differentiate the different populations. And so, if we go back and think about the challenges, one, we're not chasing the tail, working with the system as we are, we're making sure that we have alignment across the various stakeholders, and with the predictive signature, we're keeping in mind that this really needs to benefit the customer. In this case, we've identified two customers, the hospital system, which basically pays my salary, and the patient that will be the recipient of the care.
Is that your wife?
No, that's not my wife. That's some lady on Shutterstock with her cute baby, who was also on Shutterstock.
Any questions?
Yes.
Yeah, on the chasing the tail, could you name a compound or a drug where the tail was chased to good effect or bad effect?
Let me ask you this. At what point do you know that you chase the tail? Is it after the fact? Or
Well,
when do you know?
it's when, so it's looking at the fraction of a population that is impacted. So the cytochrome P450 enzymes are well known to contribute a lot to drug metabolism that impact everything from a lot of psychiatric drugs to painkillers, what have you, okay? Now, there are some very rare mutations that actually prevent metabolism of these drugs. And so these things end up being really, really pretty rare. Actually, so thinking about this, probably a a good example of this, because here's a test that really hasn't taken off, and it's for CYP2C19 with clopidogrel or Plavix. Okay, now if you actually have the mutations with 2C19, you should not be able to metabolize the drug, but it's actually such a rare mutation that the cost associated with screening everyone for that and the side effects actually tend to be relatively mild. So you can do this and it doesn't work. Another example, which was actually the first example of Genesance, was mutations in a gene for ADBR2 for albuterol response. So if you have these mutations, and you're asthmatic, you're not going to respond to albuterol. Of course, a physician can tell if you don't respond to albuterol in about 5 minutes in their office, and you don't need a genetic test to tell you that. That didn't stop us from developing the test.
I believe that where the exuberance was in 2000 with genomics, online marketing is today. Anybody who works with online big data disagree, or anyone want to talk about that? Mr. Elliot Pearson again. Yeah. Yeah. So in that video clip that we saw, right, the president was talking about the exuberance of, oh, once we map the genome, everything's cured. And in fact, that's not what actually happened.
Yeah.
And I'm saying that this is online advertising with big data today is at that level of exuberance.
Yeah, so our problem space is a little bit different because it's in our best interest to always try to find the maximum value for a particular placement. So our problem space is a little bit different than trying to solve problems for diseases and stuff. But
well, what I'm saying is, that reduction approach is that once you map the DNA right, everything's cured. Once you collect all this customer data, it's cured. But clearly, I'm guessing that's not what's happening, right? It's turning out to be harder than it actually is.
yeah, no, okay.
That's what I mean. The exuberance.
Yeah, so you would think so as far as our level of, I guess, if it was as simple as it's very easy to explain. I think Matt will actually do a great job of talking about some cool things that we do and all of that. But if it was very trivial to solve these problems, and you don't have to look at a lot of data and understand that you'll have these equations that you can come up with that have a lot of variables in order to be able to find value, and that's really that big data problem. So you actually have to put a lot of effort into it, and it's not simple. And if it was simple, everybody would be doing it. But yeah, we actually have to be very passionate about that. But we solve those problems because it was in our best interest. It's not the same when you look at it from the medical aspects. But it's great that we have that data out there, but it'll take time in the medical field for people to realize.
Cool. Are you chasing a tail? Or how would you know?
Possibly, because I actually have our existing display business. We're also getting into mobile. So right now, if you look at who's winning in mobile, it's Google. So what we're trying to do is be strong second, so we may be chasing the tail. So.
Any other questions?
Yeah.
So I have a little bit of a technical question. So what do you think about microRNA, and are we chasing the tail off that as a catch-all diagnostic?
I think that there's no such thing as a catch-all. Actually, I can remember when it was supposed to be short interfering RNAs, and it was going to be a variety of knockdowns, and it was going to be CNVs, or copy number polymorphisms, or variants. And biology just turns out to be a whole lot more complex than we believe. And I think that is why it's important to try to use whatever resources that you have, the tools that you have. Try not to be enamored too much with a tool, and let the best tools help you towards that end.